How is it to do a PhD in the Bio2Brain network?
Hello, my name is Alex, I'm from Germany and I'm currently pursuing my PhD at the University of Cambridge in the UK.
Oh, hi. My name is Dalia. I'm from Egypt and I'm pursuing my PhD. Currently, I'm working at University of Applied Sciences in Germany.
Hi, my name is Marta. I'm from Spain and I'm currently located in Florence, Italy. I work at LENS, the European Laboratory for Nonlinear Spectroscopy.
Hi, I'm Sebastian. I'm from Germany and currently working at the University Hospital in Bern, in Switzerland.
And we're performing our doctoral research in the Bio2Brain network funded by the European Union.
In the Bio2Brain network, our goal is to generate new drugs and drug delivery systems to treat various diseases of the nervous system affecting millions of people worldwide. To achieve this goal, the project is subdivided into 3 parts. The first group of PhD students aim to develop new effective treatments for specific targets in the brain, supporting regeneration. The second group of PhDs is involved in the development of new nanoparticles and microcarriers for sustained release of biologics to the nasal mucosa. Another group of PhD students is trying to understand the mechanisms of transport and distribution within the CNS, as well as the effect of these drugs in disease-mimicking animal models.
Since this task is by far too large for a single researcher to accomplish, we work as one large collaboration including PhD students from all over Europe. In Cambridge, we work on tiny antibodies, so-called nanobodies, which are supposed to bind and inactivate the molecules causing these devastating diseases. First, we design these nanobodies computationally, produce them as proteins in bacteria, purify them using liquid chromatography, and finally assess their binding to the target molecules.
At Hochschule Bibrach, we are trying to understand the mechanisms of antibody transport from the nose to the brain. We focus on interactions that take place between the Fc region of the antibody and different receptors in the airway, and how these interactions affect the fate of the antibody transport. In order to do so, we use in vitro and ex vivo models that are derived from porcine tissue due to their close resemblance to the human one.
In Florence, we try to understand the dynamics of these antibodies at the cellular level, how they move and with what receptors they interact, and also the distribution of these IgGs or antibodies in the central nervous system, which is brain and spinal cord, in order to figure out the best way to deliver these drugs to the brain. We use advanced imaging techniques like single molecule tracking, light sheet fluorescent microscopy, and confocal microscopy.
In Bern, we investigate the transport mechanism and central nervous system disposition of intranasally administered antibodies and their therapeutic efficacy in experimentally induced diseases mimicking multiple sclerosis-like pathology. For this purpose, we use in vivo models like toxic or inflammatory demyelination. With those, we have the opportunity to analyze transcriptional or expressional changes induced by the intranasally administered drugs.
A fundamental part of the Bio2Brain project as part of the European Research Programme is the interdisciplinary scientific exchange within partners of the consortium. For that, we travel around Europe to have hands-on experience on our colleagues' research centers to learn new techniques and lab culture. This will expand our scientific horizon and enrich the scientific discussion among us. But guys, not everything is going to be science. We also take advantage of our time together to discover new cities and enjoy life.